Hey guys, welcome to chapter 9. This is from DNA to protein. At this point, a lot of you guys have played that little game that I put on Edmodo for the lab, and I think that'll help you a lot as we progress through this. But this is a good chapter. This is a cool one, so I hope you enjoy it. Your book starts with a cool little story, like it always does, and for this one, it talks about a compound called ricin, RIP, which is kind of a punny sort of name because, of course, RIP we think of as rest in peace, and these guys are ribosome inactivating proteins. And so ricin is a natural protein, and it's found in the seeds of a castor oil plant. And this thing can kill an adult human if you get this protein in you, and there is no antidote to it. There's nothing we can do once it's in you. And so ricin, this little protein, is a ribosome inactivating protein. That's the RIP. And what it does is it inactivates ribosomes. And once you inactivate a ribosome, your body can't make proteins anymore, which means that everything you need proteins for is not going to work, and you're going to die. And they have this cool little story in there about an assassination in your book where a guy basically had an umbrella that was modified into a little simple gun, and he shot a BB into this guy's leg. That the BB was laced with ricin, and there was nothing they could do. They took him to the hospital, but he died 3 days later. It's nothing that can be cured. So kind of an interesting thing. There are other ribosome inactivating proteins. Shiga toxin from this guy right here, Shigella dysenteria, which is the bacterium that causes dysentery, and also one that's very similar to it that's made by E. coli, which can also make people very, very sick. And so there's your example of some very, very important proteins that affect how we make proteins. Here's your castor oil seeds. They're actually kind of pretty, that make this ricin. Here's some Jequirity beans, and these were actually, in your book, it says they were being sold on a bracelet. And a botanist recognized that the beads on the bracelet were actually these beans, and they recalled all the bracelets, but of course, they contain a ribosome inactivating protein, so that would have been terrible. There are a couple other seeds. Here's E. coli, and here's the dysentery bacteria. So there's a few different examples of where you can find some of these RIPs. So what we're going to talk about this chapter is really gene expression. Gene expression is, I guess, the act of taking a piece of DNA, the gene, and turning it into a functional protein. And then there's a lot that goes into that. And so we're going to learn about transcription and translation. Those are very, very important terms. Transcription converts the gene, or the DNA, into RNA. And you guys already know that's messenger RNA. And then translation converts information in mRNA to protein. And so you transcribe before you translate. To transcribe something means to write something down. And so you guys transcribe notes probably right now. That's transcription, right? And so if I asked you to transcribe, my name is Richard, you would write the words, my name is Richard. Now this is nice because DNA and RNA are both made of nucleotides. And so writing one thing made of nucleotides into another thing made of nucleotides, they're very, very similar. Now translation is something that looks completely different. And so here we take mRNA, which is made of nucleotides, we turn it into a protein, which is made of amino acids. So if you spoke another language, say Spanish or something like that, and I said, would you translate, my name is Richard? You would say, whatever, my name is Richard, right? Like maybe me llamo es Richard or something like that, right? So you'd take something that is information, and it's still information, but this information looks and sounds significantly different in another language. And that's where you probably know translation from in your everyday vernacular. So nucleotides to nucleotides, that's writing. Nucleotides to amino acids, that's translating. We have made something significantly different. So you transcribe before you translate. And that's how you express a gene. The reason your eyes are the color they are, the reason your hair is the color it is because of genes that have been expressed or genes that have been turned off. The combination of those is responsible for your phenotype, the way you look. So, each DNA strand consists of the 4 nucleotides. You will need to know these if you don't already. You probably do. Adenine and thymine, guanine and cytosine. A bonds to T, G bonds to C. The sequence of the bases is the genetic code, all of the information that we have, and all of the cell's RNA and the protein products that they eventually make are encoded by the DNA sequences called genes. Now, there's lots of DNA that does not code for a protein product. Before we knew what it was, we used to call it junk DNA. Now we call it non-coding DNA. And it was something that puzzled scientists for a long time. Like, a lot of your DNA, so much of your DNA, is non-coding. And we knew that DNA made proteins. But when we look at a whole genome of a little animal or a human, and we'd say, why is this huge percentage of DNA not making a protein? It's just junk. It's just there for no reason. And of course, now we know that's not the case. A lot of those DNA strands that are not coding for a protein are regulating gene expression, which is a job that is just as important as actually making a protein. So let's go with DNA to RNA first. DNA is transcribed to RNA. Remember, we transcribe before we translate. So this is transcription, same thing. So DNA is transcribed to RNA by enzymes, so proteins, that synthesize a complementary strand of RNA by reading a DNA template. So, some basic differences between DNA and RNA that you probably already know. RNA is single-stranded. RNA uses ribose sugar instead of the deoxyribose that's in DNA. That's where we get the D for DNA, deoxyribose nucleic acid, and the R in RNA, ribonucleic acid. One important difference, RNA uses uracil instead of DNA's thymine. So DNA is A, T, C, and G. RNA is A, U, C, and G. So very important difference there. Alright, so here is a DNA nucleotide. This is guanine. So you have your phosphate groups here, your deoxyribose sugar here, and then the nitrogen-containing base or nitrogenous base guanine here. Here is the RNA nucleotide guanine. Phosphates, same as the DNA. Ribose sugar, a little bit different. Nitrogenous base guanine, the same. DNA, RNA. DNA, RNA. Not a huge difference, okay? Really not a big difference. So here you go. Here's your DNA, double-stranded, the twisted ladder that we've talked about already. We have adenine bonded to thymine, guanine bonded to cytosine, the purines on top, the pyrimidines on bottom. DNA has one function, which I don't know if we can really say that, but we'll go with it. Permanently stores information. Of course, that information has a lot of other functions, but we'll say that's its function. It stores information. That's fair. Ideal situation, you're going to pass that on to your offspring. RNA, one strand. We still have our bases. Adenine. Now, we have uracil instead of thymine, but other than that, it's adenine, guanine, cytosine, and uracil. RNA is actually quite a bit more diverse than DNA. Lots of functions. We can be copies of DNA's message. We can be catalysts. We can make ribosomes out of them. All sorts of stuff. A few others even work with gene control. So again, more of that gene regulation that I told you was so important. So there are some differences there between DNA and RNA. So when we look at types of RNA, we've got a few different types you need to know about. Messenger RNA, or the mRNA. You guys probably already know this guy. He's got the information that we transcribe from the DNA. He's going to leave the nucleus and code for a protein. Ribosomal RNA, rRNA, is the main component of ribosomes. Ribosomes are the organelle which build proteins. Incredibly important stuff. Transfer RNA, or the tRNA, is going to deliver the amino acids to ribosomes. If you've already done the little lab, the online game of making a protein, that character you were trying to steer around into the ribosome, where you had to go pick the correct color ball and bring it over to the ribosome, the guy you were steering was a transfer RNA. He delivers amino acids to the ribosomes. Very important job. RNA does a lot. So our messenger RNA is what's actually going to be translated into proteins. So we take our rRNA, that's the ribosome, our tRNA, those are RNA strands that bring amino acids to the ribosome, and we translate that sequence of base triplets in our messenger RNA into a sequence of amino acids. These base triplets here, those are called a codon. Codon is important. They read 3 letters at a time, and they're going to code for a specific amino acid, and one tRNA will match that codon, and that's how you make a protein. So when it comes time for actually translating this information, we're going to carry that mRNA outside of the nucleus, and it's going to be turned into a sequence of amino acids, which are the building blocks of proteins. Results in a polypeptide chain. Our polypeptide chain is probably like, I don't know, secondary, maybe tertiary structure of protein, and that will actually fold into a protein, probably with the help of some other proteins, but that's what it's going to do. So, gene expression. Our DNA, or our genes, contains all the information needed to make all the stuff that makes you you. Well, give or take. Gene expression is a multi-step process. You have to do transcription and translation like we just talked about, and genetic information from our genes is converted into a structural or functional part of a cell or organism. And so basically we make information into stuff. We make information into a protein, and that protein could make your hair color the color it is, or it could be an antibody, where you are learning how to fight off a certain infection. Very important stuff that goes on in your body, and it all starts with information, and that information's in your cells. So gene expression, very, very important stuff. All right, so let's stop there, and we will jump into transcription, the specifics of it in our next video. So thanks for your time.